I will describe Figure 3 from the paper entitled Turning a Riboflavin-Binding Protein into a Self-Sufficient Monooxygenase by Cofactor Redesign. The author's purpose of the experiment was to create a monooxygenase that was independent from the cofactors NADP and NADPH. In order to do this, the scientists had to concentrate on cofactor redesign. A specific type of flavin, known as N5-alkylated flavins, were found to be stable in reactions involving hydrogen peroxide. The scientists decided to select the riboflavin-binding protein, known as RFBP. Combining the apo form of RFBP with different derivatives of the alkylated flavins, the scientists were able to determine the effectiveness of these artificial flavoproteins. The attachment of the flavin to the binding site of RFBP helped the scientists understand the results of the experiment. The alkylated derivatives had an opposite binding pattern than the ribotyl chain, which usually occupied the binding site of riboflavin. This could justify the opposite enantioselectivity by the flavins. The enantioselective behavior was dependent on the flavin used. The artificial construction of these flavoproteins allowed both enantiomers to be created of a set of sulfoxides. Figure 3 shows the differences between bindings of each flavin used. It shows the relationship between structure and function. The binding behavior of the flavin on the binding site of the riboflavin determined the enantioselective behavior of the flavoprotein. Lastly, the good affinity of the flavins exhibited for RFBP was illustrated in Figure 1.